All right, everybody, welcome back. We are moving right along to Module 6. So, previously, we were talking about sort of the tree of life and uh, sort of the deep life history of biology on Earth. And so, now what we're going to be doing is we're going to be diving into the branches of that tree of life and understanding what makes the different branches and what specifically helps to identify the organisms within each of these branches. And ultimately, this is talking about the organization of biology. So that's what we're going to start to get into today, looking first at prokaryotic diversity. So I thought I would start off with an interesting study from Dr. Huber. And previously, we have sort of seen these huge genetic trees that show us how different organisms are related to each other. And you saw that great video in our last course looking specifically at the structure of these trees. And what Dr. Huber has been exploring is the bacterial diversity of um, the habitats in a deep sea volcanic eruption. So the primary question or primary case study that was given in your book this week was looking at deep sea vents and understanding the diversity of life that happens in these very extreme environments. And so this is an example of assessing all the types of bacteria that are present even in these very extreme conditions. So our driving questions for this part of the section is understanding, first of all, what are the prokaryotic domains of life? Where are we on that phylogenetic tree? Secondly, we're going to be looking at the primary domains of prokaryotes and what distinguishes them. So the first domain would be bacteria. So we're going to really dive into what are the characteristics of bacteria. And secondly, what are the characteristics of archaea? So those are going to be sort of the primary questions that we're going to be exploring in the next couple lectures. So again, I just wanted to situate us where we're talking about on the genetic tree. So what we're going to be talking about this week is prokaryotic diversity. So we're looking at everything here on the left-hand side of this slide. And later we're going to be going into starting to look at eukarya, where we're starting to look at things that probably we're all more familiar with, animals, fungi, plants. But this week we're going to be looking at the diversity of prokaryotic cells, so with the two largest domains being bacteria and archaea. So these are the two that we're going to be focusing on for these few lectures. So one of the things that really distinguishes these two domains is their ability to survive in a broad spectrum of environments. And in your book, we actually looked at specifically these hydrothermal vents and all the different types of conditions that make these very difficult to survive in. There's low oxygen, extreme pH, high temperature, high water pressure. And yet, there's still these sort of dense layers of unique unicellular organisms that cover the chimney surfaces. And as we explored in another video, this is where scientists are really looking at for a potential origin of life. This is really where a lot of the primordial soup of life exists on Earth. So they're very interesting areas. And ultimately, when we're starting to think about the creation of life on Earth, we're starting to think about the very basic units of life and the most simple organisms, and those would be ultimately prokaryotes. And really, those extreme conditions also resemble ancient Earth. So not only are they likely to be the source of ancient Earth, they also mimic what Earth was like some 3.8 billion years ago. So it's an excellent study system to examine some of these questions. So a lot of what we're going to be doing in the next few lectures is really understanding what makes these different organisms unique and ultimately in their own clade on those on the tree of life. So for prokaryotic cells, there are a few things that really distinguish what makes a prokaryotic cell. And first of all, it's a unicellular organism. So we're ultimately talking about organisms that only have a single cell unit to them. They lack an internal membrane-bound organelles. So you can see that, while the cells have ribosomes and cytoplasm, they lack these complex membrane-bound organelles like endoplasmic reticulum, the Golgi complex, etc. And there's a good video that I'm going to have you watch looking at specifically what makes a prokaryotic cell a prokaryotic cell and what makes a eukaryotic cell a eukaryotic cell. Another main distinguishing factor of prokaryotic cells is that their DNA is not contained by a nucleus. So again, we're lacking those internal membrane-bound organelles, and one of those is the nucleus. So all of the transcription, translation, all of the replication is taken in a non-membrane-bound environment. Another thing that we need to think about with prokaryotic organelles is they're much smaller than what we think of it as eukaryotic cells. So this is a 2-scale eukaryotic cell, and then this is what the size of those prokaryotic cells would look like. We're really talking about a 1/10 of the diameter of a human hair, just to give you an idea of the scale of these organisms. So we're talking about smaller organisms than eukaryotic cells. And again, really one of the things that we're going to be talking about over and over again is that these organisms are really endlessly adaptive and really can thrive just about anywhere. So we saw in the Lost City in your book, we saw that these very sorry, my cat just bolted across the room like a crazy. Crepuscular madness is what my friend always calls it. So what I was saying is that, we see lots of examples of prokaryotes being able to thrive in these really adverse environments. So, we think of, these hydrothermic vents, but we also talk about, our intestines. There's no oxygen. They're a highly acidic environment. And we also see these miles underground in the salt lakes and glaciers. They're a very, very diverse organism and really can thrive in a variety of environments. So we're going to pick up next time looking specifically at bacteria, where we're going to zoom into the specific domains and really talk about what makes bacteria and archaea specific. All right. Speak soon.